Professor Stuart Hameroff received his bachelor's degree from the University of Pittsburgh and his MD degree from Hahnemann University Hospital. He has had a very distinguished career at the University of Arizona since 1975, becoming professor in the Department of Anesthesiology and Psychology and associate director of the Center for Consciousness Studies both in 1999 and emeritus professor for anesthesiology and psychology in 2003. At the very beginning of his career, while he was at Hahnemann University Hospital, cancer-related research work relating to the part played by microtubules in cell division interested Professor Hameroff and led him to speculate that they were controlled by some form of computing. It also suggested to him that part of the solution of the problem of consciousness might lie in understanding the operations of microtubules in brain cells, operations at the molecular and supramolecular level. The operations of microtubules are remarkably complex and their role pervasive in cellular operations. These facts led to the speculation that computation sufficient for consciousness might somehow be occurring there. These ideas are discussed in Professor Hameroff's first book, Ultimate Computing, published in 1987. The main substance of this book dealt with the scope for information processing in biological tissue and especially in microtubules and other parts of the cytoskeleton. In the meantime, Sir Roger Penrose had published his first book on consciousness, The Emperor's New Mind. On the basis of Gödel's incompleteness theorems, he argued that the brain could perform functions that no computer or system of algorithms could. From this, it could follow that consciousness itself might be fundamentally non-algorithmic and incapable of being modeled as a classical Turing machine type of computer. By contrast, the idea that it could be explained mechanistically was prevalent in the field of artificial intelligence at that time. Sir Roger Penrose saw the principles of quantum theory as providing an alternative process through which consciousness could arise. He further argued that this non-algorithmic process in the brain required a new form of the quantum wave reduction, later given the name objective reduction OR, which could link the brain to the fundamental space-time geometry. At this stage, he had no precise ideas as to how such a quantum process might be re-instantiated in the brain. Professor Hameroff was inspired to contact Sir Roger Penrose regarding his own theories about the mechanism of anesthesia and how it specifically targets consciousness via action on neural microtubules. The two met in 1992, and Professor Hameroff suggested that the microtubules were a good candidate for a quantum mechanism in the brain. Sir Penrose was interested in the mathematical features of the microtubule lattice, and over the next 2 years, the two collaborated in formulating the orchestrated objective reduction model of consciousness. Over the years since 1994, Professor Hameroff has been active in promoting the orchestrated OR model of consciousness through his website, conferences, and lectures. He was the lead organizer of the first Tucson Consciousness meeting in 1994 that brought together over 300 people interested in consciousness studies, including David Shammers, Christopher Koch, Bernard Bars, Roger Penrose, and Benjamin Libet. This conference is widely regarded as a landmark event within the field of consciousness studies, and bringing researchers from several disciplines together led to various useful synergies, resulting in the formation of the Center for Consciousness Studies at the University of Arizona, of which Professor Hameroff is now the director. The Center for Consciousness Studies hosts meetings on the study of consciousness every 2 years, as well as sponsors seminars on consciousness theory. All of us are eagerly waiting to hear Professor Hameroff's inaugural lecture entitled Are Microtubules Topological Quantum Computers? Update on the Penrose-Hameroff Orchestrated OR Theory. Professor Hameroff, please.
Well, let me thank Dr. Sahni and all the organizers. It's a great pleasure to be here. What I'm going to talk about today is our microtubules topological quantum computers, update on our model, which is known as orchestrated objective reduction. So the problem of consciousness is an ancient one, understanding how we have experienced conscious awareness. And we take it for granted that when we open our eyes, the world appears before us. It actually appears, at least according to modern science, inside our head. The representation of the world out there is actually inside our head, and we don't really know what's out there, technically speaking. Plato's cave is a good example of this. But trying to understand how the brain produces conscious awareness, experience, denoted here by being. And by being, I mean having experience, having awareness, as opposed to non-conscious processes. Our brains do many things non-consciously, and the brains of people under anesthesia are quite active. So being is consciousness. It's the phenomenal experience of having an experience. It's even difficult to explain, but we all know what it is. We all have it, presumably. So this is explored through neuroscientists who probe the brain by putting electrodes inside or do fMRI and so forth. Roboticists and artificial intelligence scientists who try to simulate the brain and create a computer with brain equivalents. Artists who try to capture the essence of aesthetic values in paintings and other creative forms. The physicist, Schrodinger's cat, who questioned the nature of reality and what reality actually is. The psychiatrists who probe the brain of their patients. The anesthesiologist looks suspiciously like me, who try to understand how anesthesia works. This is from a book by Sue Blackmore, who did the cartoon. The meditators, and particularly in this part of the world, who look within and do introspection. And the philosophers, going back in the West at least for a couple thousand years, and in the East even longer. So we can ask the question, what is the origin of consciousness? And I think there are three possibilities. And since we're in India, I'll start with this one, that consciousness is intrinsic to the universe as the ground of being, as Brahman or God, as suggested by Eastern spiritual approaches. And according to this, consciousness pervades a deeper level of reality, whether that's string theory or however you want to describe it. In other words, being is out in the world, out in reality. It's in the universe, and it's thought to be, in modern terms, fundamental and irreducible. Perhaps something akin to whatever gives rise to mass, spin, or charge. It's built. At least its precursors of experience are built into the universe. But you might ask, what about the brain in modern science? Because scientists want mechanisms, of course. So the most common view among scientists and Western philosophers is that during evolution, consciousness emerged from complex computation to represent reality. And that would have been a good thing for organisms, to have a picture of the world inside so they can react and deal with adversaries. So according to this view, evolution came along, and at some point during evolution, and where that point is we can talk about, consciousness emerged as a property of computation. And that's pretty much the common view in science. Now, along these lines, in Western philosophy, this is the origin of this idea that consciousness emerged from computation to represent the world, is Plato or Plotinus or several other Greek philosophers who noticed that the world out there is actually all in our head, that it's a representation. We have a representation. We don't really know the accuracy. Plato's cave is a good allegory for the fact that we don't really know that reality is as we perceive it. And so here's Plato or Plotinus, both had similar ideas, being of the tree inside the head due to light entering the eyes. So with modern times and understanding the brain and computers, the analogy and metaphor grew that here's a bunch of neurons interconnected by synapses, and variable connections among these neurons have been likened to computer switching matrices, computers of various, could be a Hopfield net or various forms of neural network, in which the brain is a computer, neuronal firings are like the bits or fundamental information units, and synapses are the switches. So that's pretty much the understanding, and in artificial intelligence they intend to, or aim to build computers along these lines that will, they say, be equivalent to the brain, to the mind, although I doubt that for reasons that I'll speak about. So the basic function of a neuron is thought to be something like this. It has many incoming dendrites that bring in inputs from synapses here, and so forth into the cell body, and these inputs are generally thought to travel along the membrane surface by ions flowing in and out, forming depolarization waves. And when they get to this part of the neuron here, the axon initiation segment, so these are all inputs, and there's one output that goes down to another brain cell here. And the inputs are integrated, and the nerve cell fires. So it converts numerous inputs, integrates them, to some threshold and fires an output. You could think of this as a analog to digital conversion. And this is the Hodgkin-Huxley neuron. It goes back 50, 60 years, in which inputs are integrated to a single output, or firing, or spike, they're called. And the spikes have been taken by AI people to be the fundamental unit of information in the brain. But I think there's a problem there. Now, for example, this work from Wolf in Germany shows that this is the predicted, these are the inputs of the cell body and dendrites that would be integrated and then fire a fairly narrow band, because the threshold is fairly narrow. So the same threshold, and it's kind of sloped because the wave propagates along these ion channels. This is the prediction for the Hodgkin-Huxley neuron. But what they find in neurons that appear to be involved in conscious processes instead is that the slope is vertical. These are all the ion channels are acting instantaneously at the same time. And also there's a very wide threshold, which means that the threshold varies from spike to spike. So the idea of a neuron as a simple integrate and fire linear system doesn't work. There's some variability in there that will alter the output, and that could involve consciousness going on inside the neuron. So if we look at a neuron, here's a biological neuron with the dendrites where inputs come in and the cell integrate, and the axon initiation segment where the threshold is, the firing occurs, and here's the output. So this is modeled in the McCullough-Pitts artificial neuron that has been used to build artificial neural networks with a number of inputs that are integrated and then output, a single output which may branch. So here's a toy neuron that shows examples of both of these with multiple inputs to three dendrites, integration, and an output. And this is so integration and fire. Integrate, fire. Integrate, fire. That's the basic idea. Although, as I said, there's something going on here that's going to influence the firing. But taking this type of neuron and putting them in a network where we have inputs coming in here and outputs going everywhere else, depending on the input, you get an output. And this is the basic idea in modern science and modern artificial intelligence upon which understanding of the brain is based. However, one might ask, where's consciousness? Why would we get conscious awareness out of this? Where's the Bing, if you will? Why would this give rise to consciousness? And the assumption among artificial intelligence computer people is that if you get complex enough, complicated enough computation, that consciousness emerges at a critical level. But no threshold has been stipulated. No critical level has been even suggested. The other problem with this is that where's the synchrony? Because we know that brain processes that correlate with consciousness are synchronized. And they're not synchronized through firing. They're synchronized through dendritic interactions. And I'll come back to that. So in summary, for this part of the talk, the problem with brain as neuronal synaptic computer, utilizing strictly classical physics, and this is kind of the understanding among most neuroscientists and philosophers, this view cannot account for conscious versus unconscious processes. There's no stipulated threshold other than to wave your arm and say, at some point consciousness emerges, but there's no stipulated threshold. Also, it forces consciousness to be an epiphenomenon, because the activity in our brains, let's say we're in a rapid conversation with two people are talking back and forth. If the person says something to me and I respond rapidly, the activity in my brain that's processing what he or she said happens after I've already responded. And because of this, the conventional understanding is that I respond unconsciously and have a false illusion of having conscious control of what I said. Therefore, consciousness is epiphenomenal, a mere helpless spectator, as Huxley said, along for the ride. And there's no free will. Free will is impossible, according to this view. And the other aspect is that there's also no possibility for non-locality, whether it's quantum entanglement or something like that. And therefore, it rules out the possibility for reincarnation, afterlife, and spirituality. And the classical physics has no room for this. It deems it impossible. And I don't think that, I think that may not be true, as we'll see. Now, perhaps to understand consciousness, we need to go to a finer scale. Now, for example, here's an example of a single cell, a paramecium. This is a unicellular organism. It swims around. If it meets an obstacle, it bounces off and goes another way. If there's a predator in the neighborhood, it leaves. If you suck it up into a capillary tube, it escapes faster and faster each time. It can learn. It can find a mate and have sex. Here are two Paramecium engaged in mating. It's a Kama Sutra position, I believe, of a single-cell Paramecium. And yet it has no synapses. It's just one cell. So the view in AI and neuroscience is that the synaptic connections give rise to the complexity in the behavior, yet here's a fairly complex behaving organism that doesn't have any synapses. And I often tell my AI colleagues and critics of our work, before you simulate a brain, simulate a single-cell paramecium. How many bits would it take to do what this guy does? Of course, we don't know whether the paramecium has consciousness. I'm not saying it does necessarily, but it might. We just don't know. So the paramecium performs its complex behaviors by structures on the outside and inside of its cell called microtubules. And these are structures, as Vishal mentioned, that are found in all animal cells, including all of our brain cells. And they make up part of what's called the cytoskeleton. So here's a cell in culture. It's a double nucleated cell, has two nuclei. The yellow is the microtubule structures, and the red are actin structures. Together, they form most of what's called the cytoskeleton. And the cytoskeleton implies that it's like the bones in our body, the skeletal structural support. Well, we think they're also the computational or intelligence processing elements inside the cell. So these are the microtubules, and they define the architecture of the cell, including neurons in terms of forming synapses and making networks. Now, when cells divide, the blue are the chromosomes. These are also immunofluorescents. The microtubules are in red, and the yellow are structures called centrioles that are the anchors of the cytoskeleton. And as Vishal mentioned, I first got interested in microtubules through studying mitosis back in the early 1970s in medical school. And all of my colleagues got interested in the genes and the genetics and the chromosomes, and I was more intrigued by how these structures knew where to go and what to do. Because if there's a problem and the chromosomes aren't divided perfectly, you can have cancer or abnormal genotypes and all kinds of problems. So these centrioles are structures that actually look like this. They're two cylinders at perpendicular angles to each other, and the cylinders are made up of 9 of these triplets of microtubules. So here are 3, 1 complete and 2 partial microtubules fused together, connected by struts to form this megacylinder, this larger barrel. And this is a very common structure in biology. It forms the basis of cilia, centrioles, flagella, very, very common. And in centrioles, there are 2 of them, and they're always at this very peculiar right angle, perpendicular to one to another. And in some cells, they're known to be photodetectors, and they're also in the retina of all of our eyes. The rods and cone cells. So all the light that gets to the rhodopsin in the back of the rod and cone cells must go through these structures. So it's interesting to note that these structures are very close to the wavelength of light and could act, it's been proposed, as some kind of optical cavity, optical resonator. Now, inside neurons, microtubules are quite rich, and here's a synapse, here's an axon, and microtubules of the axon, and the neurotransmitter vesicles that are released into the synapse. Here's the postsynaptic dendritic spine, and inside the postsynaptic dendrite, you see the microtubules connected by actin. So now most views of how the brain works and how anesthesia works also, and how consciousness is produced, is thought to be at zone A, the synapse in the membrane. But we're trying to turn attention to activities going on in zone B, inside the neuron in the microtubules, because as I hope to show you, they seem to be very adept in terms of processing information. So in the 70s and 80s, I spent a lot of time working with engineers and physicists modeling microtubules as information processing devices. So here's a microtubule. It's 25 nanometers in diameter. It can be very, very long, and axons, where they're interrupted, they can go the length of a nerve like the sciatic nerve, which could be meters long, apparently. But in dendrites, they're short and interrupted. And they're made up of these individual proteins called tubulin, which can change its shape and exist in at least two different states, actually much more than two, and possibly quantum superposition of both states. But we'll come back to the quantum business in a second. So in the 80s, I worked with physicists, for example, Steen Rasmussen at Los Alamos and Jack Tuszynski at University of Alberta, and we modeled microtubules as molecular automata. Now, a cellular automata is shown here. It's a very simple form of a computer, and this is actually the game of life, where each grid, each square in a grid can be either dead or alive, black or white, on or off, 1 or 0, however you want to put it, and then with very simple neighbor interaction rules, with each cell communicating only with its neighbor at discrete time steps, and you must have discrete time steps. With very simple rules, you get patterns that propagate and move through the lattice and can do fairly complex computation. So we applied this using the geometry of the microtubule lattice structure and the dipole coupling forces from tubulin to tubulin to model microtubules as molecular automata, and found that we get patterns that propagate through the lattice and patterns that can do information and learning. We connected two microtubules and showed that they could learn. And the propagation velocity would depend on the switching frequency the changes occurring in the 10 megahertz range. So somewhere these patterns would propagate very consistent with the velocity of action potentials. So moving through the lattice. So all this information processing can be going on inside the neuron while the neuron membrane is doing something else. So this gives a picture something like this, where we have patterns with time steps, with patterns moving through the microtubule. This is one small piece of one microtubule, so the amount of information processing is very, very large. There's also the possibility of topological computing in microtubules. Now, I'm speaking here classically. We'll get to the quantum aspect later, where instead of each individual tubulin being on or off, yes or no, a bit, we look at the pathways. And the microtubule has this interesting Fibonacci geometry with varying spiral helical pathways, 3, 5, and 8-star helices, and also along the length of the protofilament. So information could be represented, for example, by getting waves this way, or this way, or some combination, or as I'll get to later, quantum superposition of all. Let me speak about memory for a second. The best model for memory, and also how microtubule processing could relate to what's happening at the neuronal level or the synaptic level. Long-term potentiation, or LTP, is a model for memory in which if you stimulate the presynaptic neuron at a high frequency and then stop, the postsynaptic neuron becomes sensitive for a very long time. And this is thought to be how memory works, that the synapse becomes more sensitive to the same stimulus. But the proteins that make up the synaptic receptors are very transient and only last a short hours, and yet memories can last lifetimes. So the question is, where is the information stored for long-term memory, or at least for short-term memory? And one key player is a protein called CAMK2, calcium calmodulin kinase 2, which is a snowflake-shaped hexagonal protein which binds onto actin and microtubules and gets distributed through the neuron and then phosphorylates something that imparts memory. So we were curious as to whether this CAMK2 might interact with microtubules. And working with Travis Craddock and Jack Tuszynski at University of Alberta, we modeled the CAMK2. And here it is. It's this hexagonal molecule. Here it is looking on top of it, and this is sideways. Now, when it gets activated, when calcium comes in, when the nerve above it releases neurotransmitters, calcium comes into the cell and causes this to transform, and these 6 legs or feet, however you want to call them, below and above this, this association domain, forming what looks like an insect or a poodle of some sort, nanopoodle we call it. And here are the phosphorylation sites. So this is an activated form, and somehow these phosphorylate something in the cell to register memory. Well, what might that something be? We suspected, well, maybe it's microtubules, and since microtubules have a hexagonal lattice, we modeled them, and notice the scale bars here. So here is a microtubule in different perspectives, looking cross-section, and these hairs coming off are called C-termini, part of the tubulin itself, and these colored show where the tubulin gets phosphorylated. So we asked the question, how does the CAMK2 relate in terms of geometry and size to the microtubule lattice? And we found out that perfectly is the answer. The CAMK2 shown here, this is all a 10 nanometer scale bar. If you overlay it over an A lattice or a B lattice, you see that the kinase domains match up perfectly with the dimers surrounding a central dimer. So the 6 dimers here, and 6 out of 8 here in the B lattice, allowing a little curvature and extension of the legs. So these line up perfectly, and we think what's happening, and this is published, I'll give you the reference in a second, is that the CAMK2 docks with the microtubule, each of the kinase domains with one of the tubulins, and can impart information by phosphorylation. And this would show how information coming in from the synapse, from the outside world, can be phosphorylated and recorded in the microtubule lattice. So here is at the level of individual amino acids where the phosphorylation can occur. And so if we take so the point is that each CAMK2 has 6 bits. Each of the 6 kinase domains can phosphorylate a subunit or not. So that's 6 bits of information. And it's an ordered array. So an ordered array of bits is called a byte. And depending on a couple different parameters, we can record up to 5,000 bits in one little lattice neighborhood of microtubule using a trinary system. This would be a trite. So the CAMK2 can mediate information and store it in the microtubules for further processing. And if your microtubules go bad, if they fall apart, you get Alzheimer's disease. And here's an Alzheimer's neuron. You can see it's misshapen, and that's because this tau protein has fallen off and the microtubule falls apart. And when your microtubules fall apart, you can't learn, remember, and you get demented and Alzheimer's disease. There are a number of other things that go on in a microtubule. Dynein and kinesin are motor proteins that move along the microtubule and deliver synaptic precursors to the synapse. So if you want to make a synapse here, this should be a dendrite actually, these things carry things along, their cargo, and deliver them to a specific point, to a specific synapse. And the tau protein seems to tell the kinesin where to get off, what bus stop we're at, and to deliver the precursor to the local synapse. So the location of the tau needs to be registered or verified, presumably by information inherent in the microtubule. And here's the CAMK2 that we just talked about. So lots going on in the microtubule. Now, let me just say that these proteins are called microtubule-associated proteins, or MAPs, and their locations, if you just, without any inputs, form these geometric arrays here in microtubules that could be intersection points of waves propagating, or some kind of signal propagating along the microtubule lattice. Now, recently, Travis Craddock and Jack Tuszynski and I did further modeling of how microtubules might process information. And for example, one question is, well, how would the MAPs know where to bind? Because the point at which the MAP, so here's the MAP, these microtubule-associated proteins, the specific point at which they bind on a microtubule will determine the structure of the neuron and the formation of new synapses, and ultimately, in some way, consciousness. So one way, so here's the yellow might be tubulins that have been phosphorylated by KMK2. And for example, whenever, if you say wherever 3 tubulins are contiguous, then a map will bind. So by phosphorylation patterns, that tells the microtubule, which tells the maps where to bind, and that builds, it's like a building. You build the girders and then the struts, and pretty soon you have a skyscraper. And that's how neurons build through these microtubule-associated proteins. Another possibility is wherever they crisscross, wherever these pathways cross, that would be where maps bind. And also, I mentioned the kinesin marching along, and as long as it's got a phosphorylated tubulin to anchor to, it keeps going. But if there's a map there, it's told to get off, and it's going to deliver its cargo to a synapse in this neighborhood. So this could be a mechanism by which the precursors to synapses are formed and how synapses are regulated. So this is an AND gate where the inputs come in from the maps, and if you have one, it keeps on going. If you have the other, it keeps on going. If you have both, you get signal going along this protofilament. So this would be an AND gate based on map inputs. And here's an exclusive OR gate where you need, if you have one or the other, you get propagation. If you have both, you do not. Both or neither. So this would be an exclusive OR gate. The other possibility is that the inputs come in along the microtubule, and that determines, and this would be an AND gate, and that determines output along the maps. So we bring the maps into the computational system. And this would be an OR gate, I think. So the point of all this is that the brain computational capacity can be calculated in 2 ways. Conventional science, neuronal synaptic computation, the artificial intelligence crowd, AI, Singularity, would say that, who are trying to build a computer equivalent to a brain, would say that we start with 10 to the 11th neurons in a brain. Each has about 1,000 synapses at about 100 hertz. Operations gives you 10 to the 16th operations per second. So their goal to build a brain equivalent computer is to have 10 to the 16th operations per second, more or less. Now, if you go to the level of microtubules, you have the same 10 to the 11th neurons, about 10 to the 8th tubulins per neuron, at about 10 megahertz oscillation, according to Anirban, and you get about 10 to the 26th operations per second. So 10 orders of magnitude more information capacity if we go to the level of microtubules. Well, so what? How would that explain consciousness? Where's the bin? Why? This is just more computation. It doesn't explain it. Is something else required? And could that something else be quantum physics? And this is where I was at in the early 90s. I'd been spending all this time, and one day somebody said to me, let's say you're right, how would that explain consciousness? And I didn't know. So I began to read, I read Roger Penrose's book, The Emperor's New Mind, which is a marvelous book, and had all kinds of interesting things in it, most of which I didn't understand, but intuitively I felt he was onto something. So I'm not a physicist, so I'm going to give you my interpretation of quantum physics, so bear with me a second. And the basic idea is that the world reality is divided into 2 worlds. The classical world that we know very well of Newton's laws and Maxwell's equations and so forth. But at a small scale, and sometimes at large scale, but at small scale, we have the quantum world, and somehow they interact, and we don't know how. We have some ideas. We do know that in the quantum world, strange things occur. Particles can exist in multiple states or locations simultaneously. This is quantum superposition. Unified particles which become separated remain connected over distance and time. This is quantum entanglement, what Einstein called spooky action at a distance. Multiple particles can condense in quantum coherence and quantum uncertainty. To put it another way, the quantum world has multiple coexisting possibilities. The classical world is definite, deep interconnectedness, isolated, timeless, flow of time, perhaps an access to a universal mind in Eastern philosophy, and spiritual in the quantum world, but precluded in the classical world. Why don't we see quantum superpositions in our world? As most of you know, I'm sure, 100 years or so ago, the quantum pioneers found that they could have a quantum superposition. They could measure it, but it appeared as though the superposition persisted until the results were observed, measured, or consciously observed by a human. And Bohr used this to continue to do his excellent work, but it kind of put consciousness outside science. Consciousness was some strange thing that came in from the outside and caused the collapse. And Schrödinger came up with this famous thought experiment of a quantum system out here that could be both send a particle in and not send a particle in, which triggers poison, which can kill a cat. So because it's a quantum system and the particle can go through and not go through, Schrödinger pointed out that according to the Copenhagen interpretation, until you open the box and a conscious human or a conscious entity looked at the cat, it was both dead and alive at the same time. And this is the famous paradox of Schrödinger's cat. So it was intended to show how ridiculous was the Copenhagen interpretation, but we still have the problem of what happens to amplified quantum systems. How big can a quantum system be? How long will it persist? Why don't we see it in our classical world? Another view is the multiple worlds hypothesis, which is that superpositions are separations in reality. Every possibility branches off and forms a whole new universe, and we have an infinite number of parallel universes. This is the multiple worlds hypothesis, and a lot of people take this very seriously. A third possibility is decoherence, that quantum states are eroded by any interaction with the classical world. There's some kind of boundary, some kind of isolation, and heat is an important example of something that would cause decoherence. However, the nature of isolation is ill-defined. Why would there be any quantum systems anywhere? You can't really isolate. But more importantly, in recent years, it's been known for many years that quantum states may be pumped by a laser. To build a quantum computer in a lab, most people get rid of the heat by cooling it down to near absolute zero. But you can also have a quantum state by pumping it with a laser, like this thing. And also, more relevant to this talk is that evidence in recent years in photosynthesis and now in microtubules suggests functional quantum states in warm biology, that somehow biology can use the thermal environment to form quantum states which resist decoherence. We'll come back to that point. The fourth possibility for the fate of quantum superpositions is objective reduction, the most famous example of which is Roger Penrose, called objective reduction, in which an objective threshold causes reduction, or a self-collapse of the wave function. So what Roger said was that quantum superpositions which avoid decoherence long enough will grow to meet objective threshold, but rather than continue and form a new universe, when they reach this threshold given by E equals h over t, they will undergo spontaneous reduction or self-collapse, termed objective reduction or OR. I'll explain E equals h over t in a second. So according to Einstein, mass is equivalent to curvature. And he talked about it at large scales, but Roger applied it to small scales and used these cartoon space-time sheets as examples. So imagine we take four-dimensional space-time and compress 3 dimensions in one along this axis and time in this axis. So a particle in one place, say this pointer over here, would be curvature, say, into the screen, and the pointer over here will be curvature out of the screen. And so moving it back and forth is like its underlying space-time geometry flipping back and forth between two curvatures. Now, a superposition, according to Penrose, is curvature in both directions at the same time. So that the pointers here, and the space-time geometry is curved in both directions at the same time, and you have a bubble or separation in the structure of reality. Now, in the multiple worlds view, each of these sheets would go off and form a new universe, but Penrose said that these separations are unstable, and after a time t, given by E equals h over t, will spontaneously reduce to one curvature or the other. objective reduction, or OR. And he said, this is the real kicker, that when this happens, that's a moment of consciousness. And the reason for this takes up a lot of his book, The Emperor's New Mind, arguing through Gödel's theorem and so forth. So I won't go into that. I'll just say it's axiomatic that when this E equals h over t occurs, and this bubble or separation chooses either this curvature or that curvature, there's a moment of conscious awareness. Bing, happens. So what Roger said was that consciousness is an actual physical process, a sequence of quantum state reductions connected by E equals h over t to irreducible conscious precursors are inherent in fundamental space-time geometry. That the precursors of consciousness are built into the universe, very much like Eastern philosophy says. So you could say that consciousness is happening on the edge between the quantum and classical worlds. So this raises a third possibility for the origin of consciousness, that biology evolved the connection to consciousness or its precursors intrinsic to the universe. That consciousness is built into the universe like spin, mass, or charge, qualia, whatever you want to call the precursor of consciousness. But the question is how. And as Vishal said, Roger didn't have a good quantum computer in the brain, and I suggested microtubules. So we came up with the Penrose-Hameroff OR theory of quantum computations in microtubule connected to fundamental space-time geometry. And the basic idea, I told you about microtubules before. If you look inside a neuron and look at individual microtubules and then look at an individual tubulin, it forms a qubit, a protein qubit. It can be two states, two alternative states, or a superposition of both states. Now the size difference, the mechanical shape change turns out not to matter, and I'll come back to that point, because that would require and generate heat. But this is the basic idea. And we came up with this, we first started really fleshing this out in a hike in 1994 after the first Tucson conference at the Grand Canyon. There's Roger, there's me, this is David Chalmers and Jeff Tollickson and a number of other people in 94 when we first began to come up with this idea. So let's look into this a little bit more carefully. If you look at the idea of switching the protein, this was my job, being the medical person, of explaining how the protein switching could be a quantum effect. And I came up with the notion of these hydrophobic pockets, they're called, of regions within the protein that are nonpolar, shielded from the aqueous environment. So when people complain that the brain's too warm, wet, and noisy, it's not wet in there. There's no water. It's hydrophobic. Water is not allowed. Now, why hydrophobic pockets? Well, why nonpolar regions in the brain? Well, I knew from studying how anesthesia works that it looked like consciousness occurred in nonpolar hydrophobic regions within the brain. And this goes back to Meyer and Overton at the turn of the 20th century, who looked at a bunch of gases and asked how good of an anesthetic they were, whether it's tadpoles flipping over or mice or whatever, they could put them to sleep, and they looked at the potency of the anesthetic. So in terms of MAC, so the lower the MAC, mean alveolar concentration, the more potent down here, and what they found was this amazingly straight line over many orders of magnitude for their solubility in olive oil. So they were looking for some physical parameter that would correlate with the potency of the anesthetic, and what they found was solubility in olive oil. Well, why olive oil? Well, if you look at the human body, and if you were to grind it up, say grind me up and then distribute my remains, whatever, in different solvents, you find that they would distribute in different solvent spaces, and this is polarity here. So water, aqueous polar material would be out here, and here you have various other solubility media. And 3, 4, and 5, the aromatic rings, the nonpolar rings, are where xylene, toluene, and benzene are where anesthetics bind, and therefore where consciousness occurs, we would argue. So if you take what Meyer-Overton is telling us, it points to consciousness and anesthesia acting in this particular region, but it's essentially like olive oil or benzene. Now, benzene is plentiful in at least some parts of the world, but in bulk form, it's gasoline and petroleum. But in individual or in flat sheets, it's graphene, which can have quantum properties. And an individual benzene which has electron clouds, delocalized pi electron clouds, because you have 3 extra double bonds, can either be seen to flip back and forth between 2 states or to be completely delocalized in a single benzene. But when you get 2 of them nearby, they attract each other by these van der Waals-London forces, and these form a dipole and repel the electrons here, and you get dipoles like 2 little bar magnets that attract, and they can oscillate back and forth. So here we have London force dipole oscillations, and anesthetics get in and form their own London forces, which prevent these oscillations, which appear to be necessary for consciousness. So amphipathic biomolecules have a nonpolar end and a polar end, and in proteins, the nonpolar ends tend to coalesce in the middle and form a hydrophobic pocket, shown here with an anesthetic sitting in it. So these are small regions within proteins which somehow seem to control the state of the protein. So the model we came up with was that there's, in this case, 1 hydrophobic pocket, turns out there could be many, where the London forces are aligned, and as it flips back and forth, the protein changes shape and changes state. Now, it turns out this large conformational size change isn't necessary. But we also proposed that you could have quantum superposition of both states, leading to quantum computing. Recently, we've modified our model to say that you don't need the conformational state change, that all you need is the electronic dipole forces oscillating back and forth at 8 megahertz that was first discovered by Pokorny and then validated by Anirban Bandyapati. Now, looking more closely at this, if you look at one individual tubulin doing molecular modeling, this was done by Travis Craddock and Jack Jasinski and I. So here's one tubulin, and what we've modeled here are the benzene-like rings, actually in phenylalanine, tyrosine, and tryptophan. And you see there's quite a few. So instead of having 1 hydrophobic pocket, we have these sort of channels or grooves. And the green are where anesthetics bind with high affinity. So you can see they're quite near, in some cases directly in these hydrophobic regions. And we think that these hydrophobic grooves may be very important. So if we blow one of these up and just take a closer look, it also looks like there could be a helical spiral going on in here. And this pathway would give you something like this in a tubulin. And here is what's happening in photosynthesis. We know that this gives rise to some kind of coherence, or apparently quantum coherence, by propagation of excitation or electronic excitation from these chromophore to chromophore. And we're suggesting that the aromatic amino acids act in the same way, and you can have propagation along this pathway here. Now, what that would give you would be something like topological quantum computing. Because if you go back to the microtubule A lattice and align these up, so you're having a continuous array of these nonpolar amino acids where you can have a quantum activity. They align and follow this helical pathway along the microtubule, and therefore you get macroscopic quantum states because these things can be microns long. Microtubules can, or even longer in axons. So you have, by virtue of the alignment here, aligning with everything else, macroscopic quantum fractals, quantum spirals that may also be fractals, since it looks like we may have helical arrangements here. So this is the argument for the topological quantum computing that we first suggested roughly in 2002, where the different pathways could be the bits, and you could have a superposition. I don't have it drawn here, of all these possibilities, and when objective reduction occurs, one is chosen, and then that determines the behavior of the microtubule and the neuron. Where what he did was he took an individual microtubule and put 4 electrodes on it in an amazing experimental achievement. And here's the microtubule here with the four electrodes. And on 2 of these, he applied radio frequency alternating current, megahertz and kilohertz. So this is applied, the frequency applied here to 2 of these electrodes. And then this shows the resistance measured from the other 2 electrodes. And what he found was that at specific resonant frequency, about 12 kilohertz, about 20 megahertz, about 10 and 8 megahertz, you get this huge drop in resistance and increase in conductance, which may be quantum coherence. And he'll talk about that. I'm not sure you can technically call it quantum, but there's something funny going on here. It seems to be something like quantum coherence happening in microtubules at specific resonant frequencies. So they have to be resonating at the right frequency to have this quantum effect. So to kind of back up a little bit and talk about OR, I think Vishal showed this slide that if you get a quantum superposition buildup by equals h over t, you get, so here's the quantum computing, self-collapse, objective reduction, bing, consciousness happens. And each superposition tubulin chooses a classical state, and that's the outcome of the quantum computation, and that will go along to trigger axonal firing, to regulate synapses, to do what neurons do, and also manifest consciousness. So again, we have a sequence of quantum steps. Bing is reached here by equals h over t, and the process starts again. I'll just show a quick video here.
Within the brain is a forest of neurons. The principal way brain cells communicate with each other is by exchanging chemicals. That's the large-scale view which all biologists understand. But in the neurons is a denser jungle of tiny structures called microtubules. Constantly flickering when you're conscious, they stop functioning under anesthetic. Hameroff concluded these microtubules must be an essential element in what creates consciousness. They create a secluded environment where quantum events could occur. He believes it's due to these quantum events that consciousness occurs at all.
Sounds more impressive with a British accent, I found. So one might ask the question, well, if you have quantum states in one neuron, how do you get it to other neurons? And we proposed gap junctions. Actually, there's a paper here at this conference about gap junctions and synchrony, which causes synchrony, that the quantum states in one can spread through gap junctions, and you can get quantum states in microtubules throughout the brain that way. And this gave rise to kind of a spin-off called the conscious pilot, where through gap junction connections at gamma synchrony, the consciousness literally moves around the brain. And my colleague Mark Ebner in Germany and I have a paper out on this, doing extensive computer simulation of this type of thing in an artificial system. So this could be useful for agency in a computational system as well. One other point I want to make is that if you record from the cell body and the distal dendrite of the same neuron separated by 50 microns, the internal electric field potential correlates. And this is, if this were done by an action potential or a membrane effect, this would be very, very slow. And yet there's instantaneous correlation. This could be quantum entanglement. I suppose it could be an electromagnetic field. But you have the other problem of long-range quantum, long-range coherence in the brain, front to back, exactly synchronized, which is difficult to explain through classical mechanisms and could be some kind of long-range dipole correlation or quantum correlation. So let's just talk about E equals h over t. E is the gravitational self-energy of the superposition, the degree of mass space-time geometry separated from itself. So the larger the mass, or the more the space-time separation. And we can calculate that. I'll show you that in a second. H bar is Planck's constant over 2 pi, and t is the time at which OR, conscious moment, occurs. So, for example, the gravitational self-energy can be, for a given mass, can be given by gm squared over a sub c, where g is the gravitational constant, m is the mass, and a sub c is the separation distance, which it turns out is only the diameter of a carbon nucleus. So a superposition tubulin, a whole protein, need only be separated from itself by the diameter of a single carbon nucleus. So it's very, very slight, but you're still separating space-time, so that's why it's important. 2.5 femtometers. So for t equals 25 milliseconds, E is quantum superposition of about 2 times 10 to the 10th tubulins. And if you assume 1% of tubulins involved, this gives around 20,000 neurons for each 25 millisecond conscious moment. Now, I mentioned earlier about evolution and where consciousness arose during evolution. And here's the Earth being formed 4.5 billion years ago. The cytoskeleton came in here, according to Margulis's theory of endosymbiosis. And some people would say consciousness happened only recently in humans or in apes, or we don't really know. But what's interesting is that the Cambrian evolutionary explosion about 540 million years ago, there was a very brief 10 million year burst of evolutionary activity, and all the animal phyla developed very rapidly. And if you look at the organisms that were present at the beginning, you get these tiny worms and urchins, and we have the same organisms today. For example, this one, Actinosphaerium. And if you did a cross-section of one of those axonemes, you see microtubules. So each of those protruding things is a double helix of individual microtubules. Each of these is one microtubule in cross-section. And this is about 10 to the 9th tubulin. So this axoneme, according to Orko-R, could have a moment of consciousness. And it could be that at the beginning of the Cambrian evolutionary explosion, these organisms had simple conscious awareness, which accelerated evolution. It certainly would have been advantageous to have consciousness. So finally, I want to wrap up with talking about the space-time connection. And we heard several things about string theory. And the basic idea is that this Bing is actually happening at the level of space-time geometry. And the idea is that we have conscious experience. The conventional view would be that the redness of the rose is a pattern in the brain that gives rise to Bing. But there are problems with that, as I mentioned. The other possibility is that Bing is in the universe, is in the redness of the rose per se, which is reproduced in our brain due to the processes I've described. So that the conscious experience is due to a particular configuration of space-time geometry in the brain, entangled with what's out there in the real world, perhaps. Well, the idea has been around for a long time in various cultures that mind and intelligence are woven into the fabric of the universe. In the Hindu tradition, it's called the Akashic record, where information is embedded there. And if you look at space-time geometry, this is from Brian Greene's book, and he's a string theorist, and I've actually turned this upside down, showing multiple scales going down and down and down. This is the Planck scale, where there's information laid in there. It seems smooth on the way down. And there's also evidence recently that there may be some kind of fractal hierarchy in the universe coming from gravity wave experiments, so that this information, and there's also 1/f fractal activity in the brain, so that information repeats at multiple scales in a self-similar way. So that the information, even the Planck scale, can repeat at discrete levels and actually reach biological levels. And what does the Planck scale look like? Well, Calabi-Yau's string theory would look something like this. Each of these edges would be like a Planck length. Loop quantum gravity spin networks give something like this, somewhat similar, actually. So we have this geometry at the most basic level. And underlying this, according to Roger, is twisters, which are also fractal in that these sheaves, cohomologous sheaves, seem to repeat. This would underlie, this smooth continuous would underlie the discrete. So the E equals h over t, I mentioned scale-free dynamics, and that this can go from the brain all the way down to the basic level of the universe. Now, in the brain, we have these different frequencies of activities. For example, Regal showed default mode switching at about every 10 seconds. We have EEG at about 10 hertz to 100 hertz. Anirban, Bandyapati, and others have shown 10 kilohertz. Pokorny and Bandyapati have shown 10 megahertz. Jibu and Yasui talked about gigahertz and microtubules, and also terahertz. So if we use E equals h over t and put this in terms of the mass or the number of tubulins, right about here we run out of brain, because there's only about 10 to the 19th and 10 to the 20th tubulins. So even though people say we're only using a small part of our brain, that may not, when we go to these higher levels and deeper levels. And what I'm suggesting is that these deeper levels may be altered states, meditative states, enlightened states even. And so E gets larger and T gets shorter as we go deeper in scale. and could even leave the brain, because down here you're getting enough energy, you wouldn't necessarily have to need the biology. You could have activity, in non-local activity in space-time geometry. And this is actually similar to something in a Hindu tradition of lokas, or different astral planes. And maybe that actually happens, and that E equals H over T is taking us to deeper levels. Or as the Beatles said famously in a song, the deeper you go, the higher you fly, the higher you fly, the deeper you go. As you go deeper into space-time geometry, you have more intense experience. Perhaps something like this. Perhaps even accounting for out-of-body experiences and end-of-life brain activity. It turns out that if you look at EEG activity as somebody dies, there's this burst of gamma synchrony activity. This has been shown repeatedly, and it's something that's actively under study. But to wrap up with some skeptical criticisms, Max Tegmark, for example, said the brain is too warm and wet for delicate quantum effects. He published this equation for decoherence, where, interestingly, temperature was in the numerator, which would seem to go the opposite direction. But basically, what he calculated superposition of a microtubule at 10 to the minus 13 seconds, but his superposition separation was 24 nanometers, which is ours is femtometers. So that alone was like seven orders of magnitude. So we published in the same journal, PhysRev-E, and calculated down to 10 to the minus 1 second. But again, that's just calculations. And other objections came from the Australian group, and they attempted to invalidate us by saying that the electron cloud of a hydrophobic group can't oscillate, but that's only true for single benzene, so they missed the point entirely. And they also said we need GTP hydrolysis, which we don't, so I'll just skip over that. And the final objection was answered by Anirban in his recent paper that's coming out in Nature Material Science, so I'll skip over that. And so again, this is the results he found that show something like a quantum coherence effect in microtubules. If it's true that microtubules have resonant frequency in the megahertz, could that be used practical application? Well, it turns out that a common clinical tool we use called the ultrasound device, which is used for imaging, puts out megahertz, and it bounces off and we get an image. And so we did an experiment where we put ultrasound into the brain. I tried it first, and it actually felt quite pleasant. And we showed an improvement of mood in chronic pain patients. So it could be that megahertz, putting megahertz resonance into the brain might promote microtubule activity and treat a number of different diseases. So I want to conclude and just mention Vishal kindly mentioned the conference we're having. We have it every 2 years, and it's coming up April 2012 at a resort outside Tucson. This is a network of saguaro cactuses looking like a neural network, and the website is consciousness.arizona.edu. David Chalmers and I organized the conference. And so let me conclude by saying that, number one, consciousness, I think, and this is a philosophical viewpoint, is intrinsic to the universe, and biology evolved to connect to this proto-conscious quantum information in space-time geometry. Our theory describes consciousness as topological quantum computing in brain microtubules, each terminated by a Penrose objective reduction, and thus a process occurring in space-time geometry. Therapies such as transcranial ultrasound may be aimed at microtubule megahertz may be useful in neurological and psychiatric disorders. And according to our theory, consciousness can be non-local and exist at different fractal-like scales in space-time geometry, possibly accounting for so-called mystical experiences, which may turn out to have some validity. Thank you very much.
Any questions?
Let's say in the conscious world, ultimately humans have to make, let's say, a decision as they try to reach a goal. And it sort of boils down to two things. One is, do you sort of act on your own, or do you cooperate with others? It was sort of as exemplified in Prisoner's Dilemma. And I have two questions. One is, are you looking at research that links us to a higher level of consciousness, such as decision-making? Or if that's not the case, intuitively, do you think this will be possible to go right into that basic level?
Yeah, well, if you believe that there's information, Roger called it platonic values embedded in space-time geometry. Platonic values, the Hindu version would be the Akashic record of ethical and aesthetic values actually embedded in the universe. It could be that this is what's influencing us, and because in other forms of quantum mechanics, the choices of a superposition which it chooses is random. But Roger had this idea that the choice is not random, but is influenced by these platonic values. And it could be that you could call this following the way of the Tao, or your higher power, or something like that, in a mystical, spiritual sense, that we could be influenced by positive ethical, aesthetic values embedded in the universe. I'm not sure if you were asking that, or if you were asking about cooperativity among people. And it's certainly true that if you want nonverbal or non-local communication, you need something like quantum mechanics entanglement between people. And I think there's actually fairly good evidence in the so-called parapsychology literature, but the problem is because it's outside of Western science, it's put over there in parapsychology. And for example, one of the talks at the upcoming Tucson conference is by a Cornell psychologist named Daryl Bem, who, there's been these ideas of backward time effects in the brain going back to Ben Libet, and repeated many times in the parapsychology literature, where if you put images up on the screen, you react before the image occurs. And Daryl Bem did 9 experiments, all of which showed this backward time effect. And, he published it in a mainstream psychology literature after a lot of, difficult review processing. And so I give him a lot of credit. So people are still, questioning him. But he's going to present, we're going to have people debate. So it could be that we're getting information from the near future, actually. How far in the future, I don't know. But Libet's experiment showed 500 milliseconds. Now, if that's true, then that gets us out of the problem of being epiphenomenal. Because I mentioned before that if you and I are talking rapidly at a cocktail party or something, and you say something and I answer right back, the activity in my brain processing what you said happens after I've already spoken. But if there's backward time effects, the processing can send it backwards in time, and I can respond consciously. So, and in quantum physics, the flow of time doesn't necessarily, you don't need an arrow of time or a flow of time, and it could be that consciousness creates this apparent flow of time. And so we're getting help, we're getting information, perhaps from the universe and perhaps from the near future, and perhaps from other people as well.
Thank you. You're welcome. I have a small question. How do you explain attention? What phenomena is this attention? The gong rings. Somebody asks me, I'm concentrating on a book, and somebody says, has the bell gone? I say, well, I didn't pay attention to it.
Yeah.
So how do you explain attention in this?
Right. Yeah, that's a good question. And actually, we're having another session at the conference. Can you have attention without awareness? And it depends on who you believe, but it looks like you can have that attention and consciousness are doubly dissociable. You can have attention with or without consciousness, and you have consciousness with or without attention. So, for example, if you're driving, particularly on these roads, and you're daydreaming, maybe you shouldn't daydream driving in, but when I'm driving in Tucson and I'm thinking about the cases I'm going to do in the hospital that day, or why my team lost the night before, or something else, I'm still driving quite well. So the car will be in my attention, but I'm not necessarily conscious of it. And yet, if something happens, if a horn honks, then I become conscious of it. So attention and consciousness are not necessarily the same thing. Certainly they can coincide, but they need not coincide. And computer systems can have attention, can have things that warrant attention, that trigger alarms. We have a lot of alarms in anesthesia, for example. So the basic point is that consciousness and attention are doubly dissociable. You can have either with or without the other.